You're welcome at Medical Sciences by Naftali Muhumza. And in this session, as we are continuing with enzyme-linked immunosorbents assay, what we are calling ELISA. In the previous video, we have finished direct versus indirect ELISA. And in this, we want to discuss about sandwich and competitive ELISA. And some of you, I know you have eaten sandwiches, whereby you find something bound in between, and want to see how sandwich differ from competitive ELISA. And within sandwich ELISA, and as we begin, we always remind you to subscribe, like the video. What is the importance of liking the video? Whenever you like the video, you give us the confidence that we may call you are appreciating our work, and we get the energy to continue serving you. So whenever you like, you give us the energy. So we urge you to subscribe and like the video. Then, in case there is a comment, share for the betterment of our learning. And in sandwich ELISA, we get a microtiter well which is pre-coated with antigen. And as to remind you that ELISA, it deals with antigen-antibody reactions. And these antigen-antibody reactions, we use antibodies that are monoclonal. So what we are seeing in sandwich, the microtiter well is pre-coated. So it is pre-coated with monoclonal antibodies. So we get a microtiter well already pre-coated with monoclonal antibodies, specific monoclonal. If it is for determination of maybe determination of follicle stimulating hormone, it is specific to follicle stimulating hormone. If it is for determination of HIV, it is specific to HIV-1. So it is specific monoclonal antibody. So after getting a microtiter well which is already pre-coated, what we do, the first step is we are going to add, at the same time, we see we add both unlabeled, but here we are adding the unlabeled antigen. We add the sample, and this unlabeled antigen is in the sample. We add unlabeled antigen in the serum sample. So this serum sample contains the antigen which we don't know the quantity, but we are using a specific monoclonal antibody to capture the antigen in the sample which we want to determine the concentration. And after adding this sample, we see if the antigens are present which are specific to the paratopes on the antibody, which is pre-coated, they come and bind. We see them binding at the paratope of the antibodies. But remember, they are in excess, some will not get where to bind. So these ones bind to the paratopes, and these ones, because they are in excess, they fail to get where to bind. So our next procedure after adding these unlabeled antigens, the next thing is to wash. We wash with the buffer, especially phosphate buffered saline, to remove unbound antigens from the sample. So by the time we go here, we shall find we are remaining with only bound antigens from the sample, which we do not know. So here we add, then we intubate, and during incubation, we see them reacting to form what we call antigen-antibody complex is formed. This antibody-antigen complex, after incubation at room temperature, we wash to remove the excess and bound antigen, remaining with the only bound. After remaining with the only bound, the next step here is that we are going to add enzyme linked. So in this step, after washing, we come and add labeled monoclonal antibody in the reagent. This one was in the sample. This is the antibody in the reagent. So when we have already pre-coated the microtiter well with a monoclonal antigen, with a monoclonal antibody, then there is already an antigen binding from the sample. So the labeled antibody comes and binds on the epitope of the antigen from the sample. So if this is labeled, it comes and binds. We add and intubate. So they come and they bind, and we remember these enzyme-linked antibodies. The common enzyme used is horseradish peroxidase. They come and bind, but they are in excess. Some will lack where to bind. So we find some excess not binding. So during intubation, it binds, and after adding this labeled antibody, it binds or it sandwiches. So we write a statement here saying that on addition of the enzyme-linked monoclonal antibody, it sandwiches, or the antigen in the sample. The antigen in the sample is sandwiched in between the two antibodies. In between the two antibodies, that is the pre-coated and the enzyme linked. So this is why we call it sandwich ELISA. That we are saying we have an antibody pre-coated and another enzyme linked antibody, and it is sandwiching, or it is joining, it is binding the antigen in between, and that is what we call sandwiching. So that is what we call sandwich. So on addition of the enzyme-linked monoclonal antibody, and if the sample contained antigens that are specific to the paratopes of the pre-coated antigen, we see them sandwiching the antigen in between the two antibodies. And after this step, we still wash to remove this excess and bound. We wash to remove excess and bound antibodies. And after washing, remember this enzyme requires a substrate. We add the substrate, and the substrate we commonly use, or the chromogen, is tetramethylbenzidine. When we have used the CRP, and when we add this substrate, we see this HRP, which is on the enzyme converting TMB, that is the substrate, into a blue-colored product. It forms a blue product during intubation. We add a substrate and intubate, and it is during intubation that results to formation of a blue color. The blue colored solution is produced during intubation at a specified time by manufacturer. Sometimes we say 20 or 30, but it depends on the manufacturer. After intubating and the blue color has been formed, the next step is we add the stop solution to terminate the reaction. And the stop solution commonly used is 1-normal sulfuric acid. And when we add it, it changes the color from blue to yellow. So in acidic media, this indicator in the tetramethylbenzidine always changes to yellow color. And this yellow color formed, the yellow colored solution formed, is directly proportional to the amount of antigen in the sample. And this amount of antigen in the sample is measured spectrophotometrically at 450 nanometer wavelength. So this is the principle we can talk about of sandwich ELISA. So under sandwich ELISA, the first thing, we have a pre-coated microtiter wave. The microtiter wave is pre-coated with the antibody, with the monoclonal antibody. So we add the sample containing the antigen, which we don't know. And if they are compatible, we see them binding to the paratopes of the antibody pre-coated. Then we wash to remove the excess. And after washing to remove the excess, we now add enzyme-linked monoclonal antibody. And if this enzyme-linked monoclonal antibody, there is already bound antigens on the antibody. It binds on the epitopes of the antigen in the sample, sandwiching the antigen in the sample in between the two antibodies during incubation. So it is sandwiched. Then after incubation, we wash to remove the excess. And after removing the excess, we add the food for the enzyme, that is the substrate, tetramethylbenzene, which is during incubation, it is catalyzed by HLRP, leading to production of a blue-colored product. And this blue-colored product is stopped by addition of a stop solution, 1 normal sulfuric acid, leading to formation of a yellow color. And this yellow color is the one that is determined spectrophotometrically using 450 nanometer wavelength, and the intensity is directly proportional to the amount of the antigen in the sample. As we know that this sandwich is used to detect antigen in the sample. So it detects the antigen, and it is the most specific and sensitive type of ELISA. So this is what we can talk about with sandwich. Then in a competitive ELISA, the word is there is competition. So in this competitive ELISA, the principle of it is that we have the microtiter is pre-coated with antigen. So in this competitive ELISA, it is used to detect antibodies in the sample. It can either detect antibodies or antigen, depending which one is pre-coated. But because of our case, we have pre-coated the microtiter with the antigen, we're going to detect the antibodies in the serum sample. So if we get a microtiter with the pre-coated antigen, the step which we shall see is that we shall add both the sample, and this sample contains unlabeled antibody. The sample contains unlabeled antibody, whereas the reagent contains labeled monoclonal antibody. So we add them simultaneously. They are added at the same time. So we are seeing addition of the unlabeled in the sample and addition of the labeled antibody in the reagent simultaneously or at the same time. So they are added simultaneously or at the same time. Why are we adding them at the same time? We want to allow them to compete for the epitopes. So we are adding both enzyme-linked antibodies together with unlabeled antibodies in the sample. So these are unlabeled monoclonal antibody in the sample. These ones they are labeled and enzyme linked from the reagent. So we add them at the same time and they compete for the same binding sites on the epitope. During incubation, they compete. So we see competition during incubation. So during incubation, they compete for the same epitopes on the antigen. So they are competing during incubation for the time specified by the manufacturer. And after incubation, the next step is that, of course, during the competition, we are going to see if the sample has more antibodies than the reagent. We see them out-competing. They out-compete and bind to the antigen, leading to formation of antigen-antibody complex. Why are the unlabeled from the sample binding? Because we see they are more than the labeled. So they out-compete the labeled ones. Then we shall find that these enzyme-linked ones, for them, they are redundant, lacking where to bind, because they were out-competed. But as As you see, still there are some excess of also the unlabeled from the sample, which are also not binding because of the limited number of the epitopes on the antigen. So the next step from here is we need to wash. Why are we washing? Is to remove the unbound antibodies, both enzyme linked and unlabeled. So we wash with the buffer to remove the unbound, and after washing, we shall get our microtiter well having the antigen, then we shall see the antibodies from the sample binding, and these ones which were not binding, they are washed off and we remain with the only this antigen antibody complex. And after remaining with this complex, we shall come and add our substrate tetramethylbenzidine, which will lead to production of a blue color. But in our case, we see the antigens or the antibodies in the sample outcompeted the enzyme linked. So whenever we add the TMB, we don't see any enzyme linked antigen remaining. All of them were washed. So it shall lead to the intensity of the blue color will be even sometimes not there, or it will be minimal because we see they were outcompeted during incubation. So we add leading to formation. So here we add the substrate, that is TMB, tetramethylbenzidine, leading to production of the blue color. And this blue color is terminated by adding a stop solution, that is 1 normal sulfuric acid. And this one turns the color from blue to yellow due to change in the pH of the media to yellow color. And this yellow color, it is always determined spectrophotometrically at 500 and 450 nanometer wavelength. And the intensity, normally say that the intensity here, the intensity is inversely proportional. Intensity is here not, it is not directed, it is inversely proportional. Inversely proportional to the amount of, and remember we are detecting antibodies, so it is to the amount of antibodies in the sample. So the color is inversely, it is not directly proportional because we see competition. So whenever we see the intensity is too much, meaning in the sample there was little antibodies. So meaning the labeled ones outcompeted the one in the sample and they were able to bind to form more of the blue color. So the more the blue color produced in the competitive, it means there was no antibodies in the sample. So meaning the labeled ones had to compete. But in this case, where we added the sample and it contained more unlabeled than the labeled, so during the competition, the ones in the sample which is in the black color, they outcompeted the labeled one in the red. And when we add the substrate, we are going to see, we are going to produce slightly blue color, or what we call pale blue, and it will tell us that, yeah, because this one means that the HRP was little, meaning here on washing we lost all the HRP. So the blue color produced here, the intensity was very low, implying that there was much antibody in the sample. That's why we are saying the intensity is inversely proportional. So the higher the yellow color, the higher the intensity of the yellow color, the less the amount of the antibody in the sample. And the less or the pale the blue color formed, it means the sample had much antibodies which outcompeted the labeled ones. And there was no HRP to catalyze the substrate, to form the product. That's why we are seeing the product is almost pale blue, and we conclude that the intensity is inversely proportional to the amount of antibodies in the sample. So this is what we can discuss about sandwich and competitive ELISA, whereby in sandwich we saw it, and then in competitive we have seen both antibodies being added at the same time and during incubation competing for the same binding, and the one with the higher concentration outcompetes the one with the less. So when we add the sample and the reagent, the sample containing unlabeled antibodies, then the reagent containing enzyme-linked antibodies, and they are added at the same time, they compete for the same binding epitopes, and the one with the higher concentration, which is the unlabeled in black, we see it outcompeting the one in the red, which is enzyme-linked, leading to formation of a lesser color, meaning the intensity is directly proportional to the amount of the antibodies in the sample. Thank you so much for listening. We are always privileged for you to watch from the beginning until the end. Hope you are learning so much at the Medical Sciences by Naftali Muhumuza, and may the good Lord bless you.